Nanopore sequencing is actually the first sequencing technique that is actually a physical probe of the DNA. So whereas, sort of standard sequencing by synthesis techniques actually work by recognizing the bases chemically, using the sort of natural base pairing that bases actually use. Nanopore sequencing actually works by measuring how much the bases block the pore at any point in time. We and others realized that this was actually going to be enable nanopores to see things like modified bases. So things like methylcytosine or hydroxymethylcytosine. But then also relatively easily, actually sequence DNA with expanded alphabets. Beyond just being useful for spelling things, it's actually has a large range of applications for sort of DNA technologies and synthetic biology. For a long time, we've been able to make these things with phosphoramidite, solid phase synthesis, just sort of building them one piece at a time. But people haven't really been able to sequence them with any sort of fidelity. And so we realized that the nanopore sequencing, it was actually potentially just well suited to a straightforward application to sequencing DNA with these sort of so-called XNAs, xenonucleic acids. We're able to put together a collaboration with Steve Benner's group and sort of explore what kind of sequencing is actually possible for DNA made of these XNAs. Our collaboration was able to get started off of a technology development coordinating center opportunity fund grant. This was also supported by a couple of other grants, several from the NIH. So one from the National Human Genome Research Institute, one from the Office of the Directorate, and another from the National Institute of General Medical Sciences. And then there was another grant that was from the NSF that also supported development of these bases.
Hi, I'm Chris Thomas. I'm currently a scientist at Digital Biotechnologies. I was formerly a grad student and postdoc at UW, where I contributed to this work presented here as a lead author. So here we have the non-standard bases Z, P, S, and B, part of the alternative isoinformational engineered or alien DNA sat next to standard DNA. If you squint your eyes, you might not be able to tell them apart, since alien DNA shares the same basic structure and base pairing rules with standard DNA. It forms the same B-form double helix structure, for example. Alien bases do have some key differences, though, one being that both base pairs use 3 hydrogen bonds, unlike the AT base pair in standard DNA, which only uses 2. This makes alien DNA inherently more stable than standard DNA. Another difference is this nitro group you can see on Z, which can act as a functional group, much like the side chains of some proteins, which enables some self-folding alien DNA sequences to even act as enzymes. Okay, and over to the right, we have our sequencing setup, which includes the biological nanopore MSPA embedded in a lipid bilayer membrane. The membrane separates 2 electrolyte solutions, and the nanopore provides the only conductive pathway between them. When we apply a voltage across the membrane, we establish an ion current through the pore, which we measure as our primary signal. When DNA is threaded through the nanopore by the applied voltage, it blocks some of the ion current in a way that depends on their base sequence, alien or not. We also use the HEL308 helicase coupled to the DNA to control its motion through the pore. And you can check out our group's earlier publication highlight video to see how we use the nanopore setup to study the mechanics of motor enzymes like helicases instead of for sequencing. Now, we actually apply a voltage waveform that's periodic, meaning that the electrostatic force on the DNA stretches and relaxes the polymer to floss it back and forth within the nanopore. And this occurs much faster than the helicase operates. And this is why you can see these squiggly conductance traces. Each curve segment represents multiple continuous measurements of each nucleotide within a single helicase step on a single DNA molecule. And these segments overlap one another, which allows for additional error correction. So currently, alien DNA can only be chemically synthesized, meaning that we're stuck using short, expensive oligonucleotides as training sequences to build a sequencing model. This variable voltage nanopore sequencing method I described allows us to extract a lot of information from each alien read, which we use to build an alien sequencing model from a remarkably small amount of training data, something like a couple hundred reads, which is not something that we could have done with a commercial nanopore sequencer, which would have taken orders of magnitude more data to construct. We knew previously that the MSPA nanopore is sensitive to about 4 consecutive bases at a time. So we designed a library of alien DNA strands that compactly contained every 4-base combination, plus a few extra strands with redundant 4-base sequences for quality control. We could have chosen to do this with strands containing entirely alien DNA bases, but to make our lives easier, we did flank the alien bases with standard DNA for calibration and helicase loading. So we measured all these alien sequences, assigned the signal states to the respective 4-base sequence, and thus we had a nanopore signal map for alien DNA. We tested our model by sequencing 4 pseudo-random strands of alien DNA and found we did reasonably well with a mean per base accuracy of 74%. Interestingly, when we break down performance by base, we see that we do exceedingly well calling P and Z bases, but struggle to correctly call B. Of course, there's still a lot of improvement possible, but really not bad for the world's first alien DNA sequencer. Variable voltage sequencing is very computationally involved. And so the software used, like the pipelines for analysis and data sets from the study, can all be obtained by accessing the links found in the description.
So we regularly host people visiting our lab to sort of show them how everything works. And so that's something that we're also happy to do. One of the things that I always, have to convince people of is that sequencing DNA is actually really hard. That DNA is, incredibly small, and the difference between the bases is, just a few atoms. And so what we're really doing, is in a single molecule, we're able to recognize each of these residues as we go along. And really, these sequencing accuracies are comparable to the sequencing accuracies that we saw initially with ACGT DNA. And so, really want to emphasize that this was sort of the minimal training set needed to actually enable sequencing. And as the training set grows, we're going to get a better understanding of really how to sequence these with fidelity. And really, we anticipate that sequencing accuracies will improve dramatically, just as they have with ACGT DNA. And then, more broadly, this actually adds to the understanding of how nanopore ion currents actually arise. It turns out that the physics of DNA translocation and how these bases actually affect the ion current in the pore is actually really complex. And so it's actually pretty hard from sort of first principles to actually describe what it is or like what ion current you're actually going to get for a given set of DNA bases. And so, before this, we really only had natural bases, so ACGT, and then also RNA bases, a handful of modified bases, and maybe some peptides to, sort of tease this out. But this helps pave the way for sequencing, XNAs in the future, but also other biopolymers, such as peptides and things going forward.